Okay, Miss Hafsa, thank you so much everyone to join us in today's our session. Uh, Miss Hafsa, please, you can take start.
Okay, sure, sir. Assalamu alaikum wa rahmatullahi wa barakatuh. I hope you are all doing well. Here is Hafsa Sarfraz. Recently, I had completed my master's from University of Agriculture, Faisalabad, with a core interest in plant breeding and genetics, including biotechnology in my research. Besides my educational career, I have the honor of serving as General Secretary at VitaNova International Alliance for Sciences. On behalf of VitaNova International Alliance for Sciences, I warmly welcome you all for today's insightful session. Before we begin, I would like to take a moment to acknowledge and appreciate our amazing team whose dedication made this event possible. Mr. Ghulam Murtaza, CEO and President of VitaNova International Alliance for Sciences, Dr. Muhammad Shehzad Zafar. He is serving as a Senior Vice President at VNIAS. Ms. Hafsa Sarfraz, General Secretary at VitaNova International Alliance for Sciences. Ms. Javeria Rashid, she is serving as a Social Media Manager and Project Manager at VitaNova International Alliance for Sciences. Ms. Noshaba Hassan, Assistant Manager at VNIAS, and Ms. Arusa Waris, Media Relation Officer at VitaNova International Alliance for Sciences. I really appreciate the efforts of Ms. Bisma. She is serving as the Chief Coordinator at VitaNova International Alliance for Sciences. And Ms. Nadia, she is Scientific Graphic Designer. The hard work and commitment are truly commendable. Now, moving towards our honorable guest, it is a privilege to introduce our distinguished instructor for today's session, Dr. Alveena Zehra. Dr. Alveena is currently serving as Assistant Professor Biotechnology at Jinnah University for Women, Karachi. She holds a PhD in biotechnology and a postdoctorate in molecular virology. She is also an IFBA certified professional cybersecurity IFBA global mentor and a dedicated peer mentor at VitaNova International Alliance for Sciences. Her research interests lie in bioinformatics and omics technologies, and she is passionately working on integrating these advanced tools into the field of biotechnology to foster innovation and scientific growth. We are truly honored to have her with us today, and I am sure her knowledge and expertise will add great value to this session. With that, I would now like to hand over the session to our esteemed instructor. Over to you, ma'am, Dr. Alvina. Thank you so much.
Thank you so much, Ms. Hafsa. I hope I'm audible to all of the participants and all of the organizers. I'm very humbled opportunity that has been given to me. Yes.
Yes, miss, you are audible and continue.
Okay. Thank you so much. Okay. So today our topic is omics technology in modern biotechnology. It is one of the fields like omics technology has completely revolutionized modern biotechnology, molecular biology, and the fields of genomics and transcriptomics and proteomics, etc. Since it is a very broad field, so obviously we will not be able to cover the entire field in one or two sessions, but we will try our best to have a brief introduction at least. So this session will be serving as an introductory session on omics technologies and modern biotechnology. Please, next slide. Thank you. So we will start with the word omics. The word omics comes from the suffix om, which means totality or the entire set. When we use this word omics in biology, what we are mostly referring to are large-scale studies, high-throughput studies of biological molecules, like all the DNA in the genome, all the proteins in a proteome, or all the metabolites in a metabolome. So omics forms the backbone of system biology, and system biology is all about understanding organisms as a whole rather than in isolated parts. To understand it, you can imagine reading a book. Like you can see the images on your right hand. You can imagine reading a book on a particular topic. And let's assume that it is a very comprehensive one. It is recently published, so it has very much updated information on that particular topic. It's also written by an expert of the field, so it delivers a lot of expertise to the reader. But if we compare this single book to a library that is full of books on the same topic, I feel it wouldn't be hard for all of us to realize that the person going through the entire collection will still have a broader perspective on the topic as compared to the one who reads only one book. Because the collection is nearer to the entire set of books on that topic. Omics is just like that. It's like going through a full library. So instead of just one piece of the puzzle, you are reading an entire collection that is available to you. Before we get into the details of the different approaches in omics, let's first understand the concept of the omes in biological sciences: the genome, the exome, the epigenome, the transcriptome, the metabolome, and the proteome. The genome is the complete set of genetic material in an organism, every single base pair in the DNA of a particular organism. And remember that we are not here talking about a single. We are not talking about two genes. We are not talking about three genes, but the entire set of genes, the complete set of genes in an organism, that is genome. Then the exome. I can hear my own words.
Maybe your voice is doubling.
Yes, it is. I can also hear my voice doubling here, but I cannot figure out what's happening.
There's a mic issue.
Let me mute myself and let's see if somebody else's mic is open.
I,
It seems like somebody else's mic is open.
Sir Murtaza, can you see the issue?
Okay, I hope this is sorted. So we were talking about the genome, exome, epigenome, transcriptome, metabolome, and proteome. In case of genome, we were talking that it is not about a single gene or two genes or maybe three or four genes, but the complete set of genes in an organism, that's a genome. Exome, it also has ome in it. All the protein coding regions. We know, I'm sure that you must be familiar with the protein coding region being called as an exon. We mostly talk about single exon or 3 exons in a single gene, 6 exons, 8 exons, and so on. But if we talk about the entire set of exons that are present in the entire genome, that would be exome. Exome is generally only about 1 to 2% of our DNA, but it contains the most known disease-causing mutations. Then comes epigenome. Epigenome refers to all the chemical modifications in DNA, all histone protein modifications, and these are those modifications that actually affect gene activity without changing the DNA sequence itself. Again, you would be more familiar with the term epigenetics. Epigenetics refers to the single modification, whether it's a histone modification or it's a DNA methylation or any other modification. But here we are talking about the entire set of those modifications, all histone modifications, all DNA methylations. So if we are talking about the entire set, that would be called epigenome. The transcriptome, likewise, is all the RNA molecules which are transcribed from DNA at a particular time. It again reflects which genes are actively being expressed in that genome at a particular time. The proteome is a complete set of proteins produced in a cell, produced in a tissue, produced in an organism. Likewise, metabolome represents all the small molecules and metabolites which are involved in a metabolism within a biological system. See, so you can think of each ohm as a different layer of biological information. Together, they give us a rather fuller, wider picture of biology, which is exactly why we are so much interested in these ohms, and which is exactly why omics data is so much powerful. Next, please. Here you can see two pictures. And let's imagine that you are thinking of taking a picture of this beautiful landscape, and this landscape is in front of your eyes. Now you have two possibilities. Maybe you have a normal kit lens, so you will be able to take a good shot, but that shot will not cover everything in the landscape that you are able to see. In biological sciences, this can be genetics or epigenetics, because we are studying something, but we are not studying an entire set of it. In contrast, now imagine that you have a wide angle lens, so you just step back and take a wide angle shot. And now you suddenly see the surrounding landscape, the entire landscape. This is genomics, and this is genome, this is transcriptome, if we consider the biological sciences. The same goes for proteome, metabolome, and more. So this kind of study, this kind of research gives you a more complete and more wider perspective. In biology, combining these different layers of ohms is like that wide angle view. So you don't just see the genes, you see their activity as well. You see their product as well. You see their effects also. Next, please. Next slide. So omics captures the bigger picture. Just like a wide angle lens, omics lets us see all the components of a biological system at once. And when you see all the components, it becomes possible for us to identify the hidden patterns that lie there. It becomes possible for us to detect relationship between genes. It becomes possible for us to detect relationship between different proteins. It becomes possible for us to study relationship between different metabolites that we cannot otherwise spot in isolation. So omics also enables precision solutions. It forms the foundation for personalized medicine, precision agriculture, biotech innovations. It basically transforms raw data into biological insights. We will see how, and we will see it today, inshallah. So it basically turns complex biological snapshots into some actionable knowledge. So now the types and approaches that we have in omics include genomics, transcriptomics, proteomics, and we will go through them one by one. Next slide, please. So genomics, which is the study of complete DNA content of an organism. So we can see that we can think of it as the entire genetic blueprint of an organism. When we want to explore this blueprint, we use several tools available. Like one option is Sanger sequencing, which is one of the earliest sequencing methods. You must be familiar with it, being a biotechnologist. Uh, it was developed in 1970s. It was the most widely used technique when the first genome was being sequenced. It reads DNA sequences very accurately, but Sanger sequencing is best for short fragments, at most 1,000 base pair. Not it can not go beyond that. So it is often used for targeted sequencing or not for whole genome sequencing. But still, we can say that the first genome that was completely sequenced, Sanger sequencing was the technique that was used at that time. But now, since we have other techniques, better techniques, more economically feasible techniques, we generally use those techniques instead of Sanger sequencing when we want to have whole genome sequenced. So that technique is known as next generation sequencing or NGS. It's a newer technique, it's a high throughput technology. It can read millions of DNA fragments in parallel, which is why it is also known as massively parallel sequencing technique. Next generation sequencing is very much faster as compared to Sanger. It is very much cheaper per base, and it can be used for very large scale studies. Then whole genome sequencing is actually an application of next generation sequencing. An application that determines the complete DNA sequence of a particular organism's genome in a single process. And this allows us a very comprehensive look at the genomic level, at the genomic information of that organism. Genomics has very wide range of applications. It can be used for mutation analysis, where mutations can be linked to different genetic disorders, and these mutations are identified. We perform different association studies, including genome-wide association studies, to know and to assess whether a person, because of having a particular genetic variant, whether that person is susceptible to a particular disease condition or not. It can be used to tailor treatments based on a person's genetic makeup, such as this selecting the right cancer therapy, like personalized medicine. It can also be used to identify new drug targets and predict drug response based on genetic variation. It can also help in agriculture biotechnology for allowing us to breed crops with traits like drought resistance, like pest resistance, like higher nutritional value. It can also help us track outbreaks, study how pathogens are evaluating over the period of time, and it can also help us develop targeted vaccines against those evolving and continuously evolving pathogens. Then comes transcriptomics. Transcriptomics refers to the study of all RNA transcripts. We can here recall the central dogma of molecular biology, which is that DNA gets converted to RNA, RNA gets converted to protein, and transcription is the middle step of this process, which produces messenger RNA, or we can say it produces transcript. So when we are interested in analyzing all of the transcripts which are being produced at any one time in an individual, It may be a disease state, it may be a healthy state. It may tell us which genes are switched on or off at that particular time. To study this, we generally use two main tools: RNA sequencing or RNA-seq, or microarrays, expression microarrays. RNA-seq is a very powerful next-generation sequencing method that can capture and quantify all RNA molecules, including rare transcripts and alternative splicing events. Microarrays are comparatively older, like it's an older technique, but it still is considered very useful technology where thousands of DNA probes are fixed on a particular chip to measure the expression levels of many genes simultaneously. But since RNA-seq is now being used and is available uh, to be used, and it is comparatively, as compared to microarray, it is comparatively more cheaper. So the world is now moving from microarrays to RNA-seq. Otherwise, before RNA-seq, microarrays used to be the only technique if we were interested in transcriptomics. Application of transcriptomics include gene expression profiling, which help us understand which genes are turned on, which genes are turned off in different conditions, and the discovery of disease biomarkers, where RNA patterns can signal the presence of a disease, a stage of a particular disease, such as in diagnosis of cancer. It can be very helpful. In prognosis of cancer, again, it can be very helpful. Next slide, please. Then comes proteomics, which is the study of the entire protein complement of a cell, of a tissue, or of an organism. Unlike the genome, which is relatively static, the proteome is highly dynamic. Here, by static, I mean that genome remains almost the same for the entire life period of an organism, whereas proteome keeps changing. It changes with time, it changes with environment, it changes with cellular conditions. To study proteins, we use tools such as two-dimensional gel electrophoresis and mass spectrometry. Two-dimensional gel electrophoresis separates proteins based on their size and based on their charge, and this allows us to compare protein expression patterns. Mass spectrometry is a more powerful technique, and it identifies and quantifies proteins with very high sensitivity and can also detect structural features like modifications of proteins. Applications of proteins include post-translational modification analysis, such as phosphorylation, which may regulate many signaling pathways. Applications may include drug target identification, where we can find proteins that serve as key intervention points in different diseases. A practical example is comparing proteins from healthy versus proteins from diseased tissues. This approach generally helps us identify different protein biomarkers of disease onset and disease progression and potential therapeutic targets. Beyond genomics, transcriptomics, and proteomics, there are other specialized omics fields that give us even more layers of insights into biology. They include epigenomics, metabolomics, and microbiomics. Epigenomics here looks at the chemical modification, as I mentioned earlier, chemical modifications to DNA and histone proteins, such as DNA methylation, such as histone modification, that control how genes are turned on or how genes are turned off in a particular condition without altering the DNA sequence itself. This is crucial for understanding development. This is crucial for understanding aging. This is crucial for understanding diseases, particularly diseases like cancers. Metabolomics studies the complete set of small metabolites in a cell or in a tissue, like things like sugar, like amino acids, like different signaling molecules. These small molecules and these metabolic profiles provide a snapshot of cellular activity and health at a particular time. Then comes microbiomics. Microbiomics is more widely known as microbiome studies instead of microbiomics. But since it is one of the very heavily worked upon field, uh, which is currently explored a lot, I wanted to mention these microbiome studies as microbiomics. So these explore the microbial communities living in and living on us, especially the gut microbiome. These microbes influence digestion, immunity, and even mental health. And microbiome research has actually greatly transformed how we think about human health and how we think about disease, and has largely contributed to our understanding of how influential these smaller organisms, these microbes, which reside either in us or on us, are in our health conditions or disease conditions. Now that we have looked at individual omics fields, let's take a step further into multi-omics. Multi-omics means combining two or more omics layers. Like for example, genomics plus transcriptomics, transcriptomics plus proteomics, proteomics plus metabolomics, to get a complete systems level view. And why is this so powerful? Because no single layer tells the whole story. The genome is there to show potential. The transcriptome is there to show activity. Proteins show function. Metabolites show outcome. So when we integrate them all, we can see the cause and effect relationship and capture the complexity of living systems in a much, much better way as compared to when we are studying the same fields in isolation, like we are when we are studying only genomics or only transcriptomics or only proteomics. So this integration is actually transforming research and applications from identifying better biomarkers in disease to developing precision medicine strategies and even designing stress resistant crops in agriculture. For example, in cancer research, scientists don't just look at the mutations in DNA, but now they also examine RNA expression. They also now examine protein activity together, and this gives a far richer understanding of tumor behavior and the treatment response of to any treatment that is given to that tumor patient. Next slide, please. Now that we have explored omics and even multi-omics, let's pause and focus on the raw material behind all of it, the data, omics data. Omics data is generated using high throughput technologies that we have just discussed, all of those technologies like next generation sequencing, uh, whole genome sequencing in case of genomics, RNA-seq in the case of transcriptomics, mass spectrometry maybe in the case of proteomics. So these are very high throughput technologies. So you have to understand that when omics data is generated, it is coming through all of these technologies, which include sequencing technologies, microarray technologies, mass spectrometry technology. So the result is massive. Result is very, very complex data set that captures DNA sequences, that capture RNA levels, that capture protein abundances, that capture metabolite concentrations maybe, or even the interaction between these molecules. So this data is very complex and is characterized by three key things, two to three things, key factors. First, the volume. We are talking about gigabytes here, like gigabytes to terabytes of data from a single project is generated when we are using omics technology. And it's a very huge amount of data. You need very specific uh, computers, you need very specific storage spaces when you are working with omics technology because you are working with very large volume of data. Then second is the variety. Data comes in many different formats like DNA sequences. can be found in FASTA sequence, it can be found in FASTQ, then VCF formats are there, then expression matrices are there, then different spectra like protein and metabolite spectra can be there. So when you are talking about omics data, there are a lot of different formats in which this data can exist. And you have to figure out exactly which formats you are interested in and how you are going to work with those. In short, omics data is the foundation. It is the raw material we feed into bioinformatics workflow to extract insights in systems biology, in medicine, and in biotechnology. Now, omics data can take many different forms depending on whether we are looking at DNA, whether we are looking at RNA, or we are looking at proteins or metabolites. But among all of these, the most foundational layer and the one we will focus on in detail today is genomic layer, genomic data. Because DNA is the blueprint of life, we all understand that, and nearly all omics layers ultimately depend on what is encoded in the genome. Like if we talk about the transcriptome layer, epigenome layer, proteome layer, metabolome layer, the basis is genome. So understanding how genomic data is generated, how it is processed, how it is analyzed, it provides the basic understanding and a basis for interpreting transcriptomics, basis for interpreting proteomics, metabolomics, and so on. So let's walk through the typical genomics workflow and let's see how it goes from raw DNA to meaningful biological insights. The genomics workflow can be broadly divided into two major parts: wet lab component and the dry lab component. The wet lab component begins with sample collection, and this can be anything from human tissues or human blood to microbial isolates to plant leaves or even environmental swabs. Next, once we have collected the sample, next we will move on to nucleic acid extraction, where DNA or RNA is isolated from the sample. At this stage, we will also check the quality and quantity, and we will be using a number of instruments that are available to us. We may use nanodrop, we may use qubit, we may use bioanalyzer to make sure the goal is same. Like the instruments can vary depending on the availability of them, but the goal is same, that we want to make sure that our nucleic acids are that we have extracted, they are pure and are intact. Then comes amplification and library preparation. This involves fragmenting the DNA or RNA. It involves adding adapters. It involves indexing or barcoding the samples so multiple samples can be sequenced together in a single run, and they can be easily identified later on when they are in downstream processing. Finally, we reach the sequencing step itself. Here, different technologies can be used, like Illumina sequencing, like PacBio sequencing, like Oxford Nanopore sequencing. Illumina provides short reads. Oxford Nanopore and PacBio generate long reads. Both of these reads are useful for different kinds of analysis. So depending on our goal, we decide which of the sequencing platform we are going to use, whether we are going to use Illumina or whether we are going to use Oxford Nanopore or whether PacBio would be more feasible for us. So at the end, these wet lab steps create the raw sequencing data, and we take this raw sequencing data for in the next part, next component, which is the dry lab component, and we try to make sense of this data. So the dry lab analysis begins with the raw data that we obtain after sequencing. It depends, like depending on the sequencing technology, whether we are using Illumina or PacBio and Oxford Nanopore. This comes in different formats, most likely in FASTQ files. FASTQ files are the ones that you are going to obtain, and they contain millions of reads in those files. Remember the difference between the Sanger sequencing and next generation sequencing, because this was massively parallel. We learned this. And because of the massively parallel sequencing, the raw reads that we obtain out of the sequencer, they have millions of reads in those files. Then the first step is quality control. In this step, we check for sequencing errors, we check for low quality bases, we check for adapter contamination, like the adapters that we added in the wet lab part. We look here for their possible contamination. We use tools like FastQC and Chemomatic. They are used generally to clean and to filter the data, to filter the raw data, to clean the raw data that you have obtained, and to filter out, to clean the and improve the quality of your reads. Now, next step is mapping or assembly. In mapping, what we do is that we align these reads that we have obtained to a reference genome, to a known reference genome. We generally take this strategy when we already have a reference genome of the organism available. And in case when reference genome is not available and we have nothing as a reference, so our reads can be mapped against, we use the second strategy, that is assembly. And here we try to reconstruct the genome by piecing all of the reads together. So depending on the work, depending on the availability of reference genome, or if it is not available, we choose whether we have to map our reads against the reference genome or whether we have to go for de novo assembly. Once the reads are mapped or the reads are assembled, we move to this next step, that is processing and annotation. Here, this step includes identifying genetic variants, like if there are SNPs present, if there are insertions are present, if there are deletions present. This step identifies these genetic variants. This step helps you identify structural changes if possible, and helps you annotate genes or regulatory elements to understand their function. And finally, we are able to summarize everything into plots, into results, into the visual outputs, such as genome browsers, such as heat maps, such as different variant distribution plots and tables or pathway maps. These provide insights that biologists or clinicians can use for their decision making. So wet lab generates the data, the raw data, and the dry lab component transforms that data into biologically meaningful data. Now let's talk about some sequencing file formats. Like when you are a researcher and you intend or you want to work in the field of genomics or transcriptomics or any other, you need to be familiar with a couple of file formats, sequencing file formats particularly, that you obtain as a result of sequencing, like next generation sequencing. So let's talk about them one by one. First of them that you see here are two formats, which are SAM and BAM files. SAM and BAM are very related files. SAM stands for Sequence Alignment Map, and BAM stands for Binary Alignment Map. SAM files are text-based files, and they store sequencing reads and their alignment information. Because it is text-based, you can open it in a text editor. You can easily read it out. However, these files tend to be very large. And since they are very large, they are not always efficient to store. They are not very efficient to share. To solve this issue, that they are very large and it is very difficult to share SAM files with another researcher or maybe RPI, another format has been developed which is known as BAM file, Binary Alignment Map. A BAM file contains exactly the same information as a SAM file, but it is present over there in a compressed format because it is there in a binary format, and binary format obviously, as compared to text based file, binary file, a binary based format would be a compressed version. So this makes it much smaller, and it becomes very faster for our computers to process it. However, the problem associated with BAM files is that they are not human readable. Like SAM files that we were able to open them in and we were able to see them in a text editor. If we open BAM files, they would not make any sense to us. So if we have to inspect BAM files, we need to convert them back into the SAM files, and then we can work on those SAM files. However, different bioinformatics programs and tools, they do not have this issue. Like BAM files are not human readable, but they are machine readable. They are tool readable. So if we are using different bioinformatic tools and we are actually trying to process these files over there, no difference exists between SAM and BAM files. SAM files can be processed by different bioinformatics tools. The same tools will be able to access and same tools will be able to process BAM files. So the only difference lies between human readability. Now, the next two formats, the most common sequence file formats are FASTA and FASTQ. FASTA file is the simplest way to store biological sequences. It can contain DNA, it can contain RNA, it can contain different protein sequences, and the format is very straightforward. You have a header line that starts with a greater than symbol and the sequence identifier, and this is followed by the sequence itself. FASTA files contain only the sequence and no additional information about the accuracy, about the quality of the sequence is there in FASTA format. So it's a simple sequence. However, in contrast, the FASTQ file builds on FASTA by adding quality scores for each base. Like these quality scores tell us how confident the sequencing machine was about each nucleotide call. Like if the machine called a particular base as adenine, how confidently it called it. So that is known as quality score, and quality score is uh, present in FASTQ files in addition to the sequence. So the format here looks a little bit different as compared to FASTA, and it starts with @ symbol, then the identifier, then the sequence, and then a plus separator that you can see here. And after this plus separator, it's a line full of symbols. And these symbols, like here, plus and percentage and brackets, and all of these symbols actually represent quality score. So if we are going to summarize this, in short, FASTA is sequence only file and FASTQ is sequence plus quality information. So FASTQ is the standard output for raw sequencing reads. Like most of the times, the raw reads that we obtain after sequencing, after performing sequencing, are the same FASTQ files. And FASTA is often used later in the bioinformatics pipelines and bioinformatics workflows once sequences are more cleaned and they are more assembled. Now let's move from raw sequence data formats to processed variant data. So VCF or MAF, these are the sequencing file formats which contain processed data as compared to the raw data that we were referring to previously. So VCF stands for variant call format. It is used to store genetic variants, basically places where a sample's DNA differs from a reference genome. These variants can include single nucleotide changes, they can include insertions, they can include deletions. Each sample gets its own VCF file, which is essential. Like even if you are running a running an entire batch of 96 samples. Each of those samples gets its own VCF file, which is essential if you are studying things like inherited mutations or population genetics or cancer genomics, etc. But when we have many samples, imagine we are working with an entire batch of 96 samples. Looking at one VCF file at a time can compromise our workflows, can compromise our speed, and makes it a little bit inefficient. So that's where MAF, which is mutation annotation format file, this is where it comes. In MAF file, an aggregated summary of multiple VCFs are there. Like VCF, variant call format, is there for one particular sample. And if we are working and if we are talking about an entire batch of those samples and we are trying to uh, identify different variants in all of those samples, the file format that would be most relevant to us would be mutation annotation format. So it's an aggregated summary of multiple VCFs. Instead of keeping each sample separate, it collects all the mutations across a group into one file, which makes it much easier to analyze strains such as common mutations in a cancer cohort or shared variants in a population. So, in short, VCF is a sample level variant data, and MAF is a group level summarized form of variant data. I hope it's not confusing. When it comes to data analysis, tools can be divided into two broad categories: graphic user interface based tools and command line interface based tools. GUI based tools or graphic user interface based tools come with a graphical user interface, making them more user friendly, especially for beginners. For example, maybe you may have heard about the Galaxy platform that provides a web based interface, and you just have to drag and drop uh, your sequences, your maybe fast queue sequences. You just have to drag them and drop them over there. They will be processed. You have to select and you just have to click on the respective tools that you want to use on that particular sequence, and uh, it will be run, and it will prompt you when it is completed. Then you can do the same procedure, like you can run another tool or maybe two or three tools on the same data. It is very user friendly. You just have to uh, play around using your mouse. You just have to hover over, you have to click a number of times, and the work is done. So it's a simple drag and drop interface for building analysis workflows. Remember the name Galaxy. Then there are other tools like UGene, like Genius. They also allow visualization and manipulation of sequence data. Then there are tools like IGV. They are popular genome browser, integrative genomics viewer actually. Then Cytoscape is widely used for network biology, interactive visualization. So there are a number of tools and they are all very helpful. Some of them are freely available. Some of them have membership fee and a little bit, you have to pay a little bit in order to use them. But a lot of them, and this list is not very exhaustive. I have just mentioned the most frequently used tools here, but there are a number of tools available for NGS, for genomics analysis, which are GUI based tools. On the other hand, we have CLI based tools, which are command line interface based tools. They are more flexible, they are more scalable, and they are based on scripts. Like you have to write a whole long script to perform a single task. In GUI based tools, no coding was required. They were very user friendly. Drag and drop mechanism was there. In command line tools, there is no drag and drop option. You have to learn that particular language. You have to learn particular uh, scripts. You need to be aware of particular packages. Although there are a lot of packages available, but you have to put a lot of effort. Like for beginners, GUI tools are more user friendly, and you can start off with them, and later on you can learn command line interface based tools. But command line interface tools based CLI based tools have their own advantages and benefits. They are very scalable and they are excellent when it comes to large data sets and when you have to process a number of sequences and batches after batches. uh, Command line tools are more preferable. So here you have bash. Bash is essential. Can you please go back to the same? Yes, thank you. So bash is essential for automating pipelines and managing files. Then R is there. R is mostly used for statistics, for data analysis, for visualization. There are a number of packages which are about genomics analysis, like bioconductors and many other, which are available in R, and they're comparatively very user friendly. Then comes Python. Python is another language, excellent for data wrangling, excellent for machine learning, excellent for bioinformatics analysis, but one has to put a lot of effort when they are interested in learning Python. So command line tools are very beneficial for when you want developing expertise in writing scripts. They are better when we say that they are better when we are handling large data sets. We say so because uh, when we are repeatedly performing uh, analysis one after another in GUI, it was easier to perform, but we cannot go back and trace what mistake did we do. We cannot pinpoint. We will have to perform it again and again to in order to identify what went wrong and even to recall what we did initially. However, in command line interface, since you are writing scripts here, all of those scripts would be saved. So you just have to go back, scroll upwards in that environment, and you have to identify that I wrote this script and this script resulted in so and so information. So if I need to repeat the same thing again, I need to copy the same script and I have to use the same script again. And if I need some different results, then I look at the script again and I can easily identify that I made a mistake here, exactly here in the script, and then I need to modify it, I need to correct it. So in terms of reproducibility, command line tools are more impressive. They are preferable as compared to GUI tools. But as a starting point, you can always go for GUI based tools. Next slide, please. To wrap up, let's summarize today's session. First, omics provides us with a wide angle view of biology. Instead of looking at a single gene or a single protein, we look at the whole set, the genome, transcriptome, proteome, metabolome, and beyond. Then we saw how the genomics workflow is divided into two parts: the wet lab component, where DNA and RNA is collected and prepared and sequenced, and the dry lab component, where computational tools process and interpret that data. We also discussed the importance of both type of tools, including GUI based tools, graphic user interface based tools, which are more user friendly, specifically for beginners, and CLI or command line interface based tools like Bash, like R, like Python, which are very, very powerful for large scale analysis. In terms of application, omics is now central to personalized medicine. It is central to precision agriculture, biotechnology, disease research. And when we integrate multiple omics layer in multi-omics particularly, we get an even deeper, a system level view of life. Next slide. So as we wrap up, these are some key resources and reference materials that you can explore further to deepen your understanding of omics technology. This reference list contains some textbooks which provide basic knowledge, It has some review articles which highlight current trends and tools, uh, current trends. And there are some tools, particularly here you can see the Galaxy project, like usegalaxy.org. You can set up your account over Galaxy, and there are multiple training materials available, and you can very independently work on on solving brief biological questions using this Galaxy platform, and this will help you get develop some expertise, particularly in GUI based tools in genomic analysis. Then finally, here I mentioned a Coursera course. It's a step for structured, I would say, structured self-learning, and I highly encourage self-learning because I feel that the participants of this session would at least be uh, undergoing graduation, or maybe they would be uh, students of MS or PhD level. So, individual learning, self learning is something that there is no replacement of self learning. There is no replacement of putting effort alone. So, this Coursera course is another very beneficial course. Uh, the name is written here, Researcher's Guide for Omics to Omics Data. You can start off with this course, and it will lead you to two more courses, and you will end up receiving a specialization in omics data. So it is like freely available to those who may have received the HSE subscription of Coursera, or otherwise also you can go through them. Even if you do not receive the certificate, I would encourage that you at least go through the material that is available in this course. So with this, I would like to thank all of you, and I'm open to questions if there are any questions.
Thank you so much, Dr. Alveena, for such an insightful session. And I am reading the comments from the listeners. That's amazing. There are some questions in the chat box. Can you read, or I can read for you?
It would be very helpful if you can read, because I cannot see the questions. Okay, fine. Now I can see. I
Okay,
will read.
thank you so much, the ma'am Alveena Zehra, for giving an insightful session about omics, multi-omics in modern biotechnology. It's really very insight. Thank you so much. We honor to have you. And now, Miss Hafsa, you can please, if anyone have any question, you can write your question in chat box. Miss Hafsa will read.
Yes, sure. Miss, can I read the questions?
Okay, I can see one comment that please share reference to related material links in description, please. Um, okay, I will share it. If you guys are in the WhatsApp group, I will share the detailed resources and the links to it in the WhatsApp group as well.
Here is a question. Ma'am, am I audible to you?
Yes.
Here is a question: how makes helpful in agriculture, especially in entomology field?
Omics may be helpful in agriculture, particularly in two ways in connection with entomology. Like you can perform plant omics, like plant genomics, plant metabolomics, transcriptomics, and proteomics. So you have all that plant information, right? Like all the information that's coming from plant, and that determines its susceptibility patterns towards different agents, like towards different, uh, otherwise, I would say, different. Maybe bacteria, different viruses, all of those agents, and particularly in the case of entomology, those that profile that kind of attracts different insects towards the plant. And similarly, we can also we can similarly go through uh, the genomics of those insects and what makes uh, like how the two connects together. Bilkul it would be very similar like host and pathogen connection. So we can go through the genomics like host genomics, like plant genomics as well as insect genomics, and it would be helpful in improving the overall agriculture. We can even make variations in those. Once we have that information, we can make our crops more resistant. We can work on like making those improve the resistance towards those particular pathogens or particular insects.
Miss Afsa, kindly read out question, any important question, not others.
I can read one question here, that how do sequencing errors reflected in the quality scores of fast Q files impact the accuracy of downstream analysis such as variant calling or genome assembly? We generally, when we obtain fast files, what we, as we discussed, that the first step that we perform is to analyze the quality of those fast Q files. We generally use two tools. For individual files, we can perform fast QC. It's a very simple tool. You can just download it, and if you have any fast Q uh, file, you can just insert that file, open that file up in that particular tool, and it will give you a per sequence per base call quality per base quality of that entire set of sequence. And you will see a graph, and on the top part of that graph would be green in color, and in the bottom part would be yellow, and the lowermost part is red in color. And this green is considered like, if the score, if the quality score is more than uh, 20 25, it is considered the good score. And it would fall at the top of that graph in that green color. Your individual bars, your sequence bars would be there uh, in that green region. If your sequence bars go a little lower from that green region and they are passing from green to yellow, it means that a little bit compromised quality of reads is there. And if the read is entirely there in yellow and red region, or entirely in red region, that means that it is a very poor quality and we are going to discard that particular read. We are not going to consider it further. So we use two tools, and you can just google them up. uh, fast QC is the tool name. And if we are working on the same thing, but we are working on an entire batch instead of one or two read sequences, so we can use multi QC. So fast QC and multi QC, these are two respective tools that we can use to make sense of the quality score, and to uh, make the score more comprehensible for ourselves. I hope this answered your question.
Okay. Hello, Miss. Also, am I audible?
Yes, sir, you are audible.
Am I audible to you?
Yes, sir.
Okay, ma'am. Please kindly conclude today's session.
So, I think this was the most brief introductory kind of lecture on omics technology. Omics technology is a very vast, very broad field, and it's such a broad field that it becomes very difficult for uh, to even talk about it because the tools available is so huge, is so enormous that it becomes a little tricky to explain it in simplified form so people can make sense of it and people can be inspired to take it to pursue this as a career choice. But I would highly encourage uh, every one of the participants to start thinking about working in this field. This is an area like there are limitations, particularly in those countries where resources are limited. We have a lot of issues like we want to work like very for a very on a very good topic, but sometimes it happens that our universities, our academia, our labs, they are not that much capable uh, of doing that work that we actually idealize and that we actually want to pursue. But genomics, transcriptomics, proteomics, these are those kinds of fields where even if you skip the wet lab part, and by skipping, I would mean that if we are not able to do it in our own labs, a lot of such data is publicly available. Like there are databases where genomic sequences are available. There are multiple databases where transcriptomic sequences are available, where proteomics data is available. What we can do is that we try to develop our expertise in data analysis, in analysis and bioinformatics analysis of that omics data, and that can be very, very helpful uh, on individual level for us and on a national level. It will develop our capacity, a lot of capacity, and it will give us a lot of opportunities nationally and internationally. So I would just like to end this conversation and this session uh, on the same note that try to uh, start learning about omics, particularly the dry lab part. It doesn't require a lot of stuff from you. You can simply work on this part from from the comfort of your home, from the comfort of your drawing room, uh, and you can be a very good scientist. Thank you so much.
Thank you so much, Dr. Alveena. Guys, the recording will be available on YouTube. You can watch it at any time. And if you have any queries, you can direct there. Or you can type your questions in the group, they will be answered. So, thank you so much, Dr. Alveena, for being here. Besides too much issues at the backside, but I really appreciate your presence and all other audience who are still here and listening. Thank you so much.
Thank you so much.
Okay. Allah Hafiz, everyone.
Thank you so much, everyone, and thank you so much, ma'am. We honored to have you here. In best regards, one.
To all the participants for being so patient listeners. And thank you so much for the organizers for organizing this session. Thank you.
Thank you so much, Miss, and the participants for being here till the last moment.